Yeah, it's flash the ring.
Hello, future us.
Here, probably won't be watching the
You might be studying for this test here.
Yeah. Okay. Take a deep breath. Just relax. Okay.
I'm ready.
I have 100 pens. I think I'm ready.
Okay. So please ask questions if you have. We stopped a few times to show some videos and random stuff. So, okay, your third line of defense is what we call our adaptive immunity or acquired immunity. So this is not something you're born with. Newborns don't have this yet. They have the ability to do this, to make one, to whatever, but they haven't been exposed to anything, so therefore they don't have this yet. Yes. Yeah. Like when I had my children, both times, the pediatrician was like, don't leave your house, especially with the twins. They're like, don't leave your house for, I think he told me 10 weeks with the twins, and it was 8 weeks with Marie.
Like how people sometimes like bring their puppies outside and like they play outside and they're just like dying with really young because they're like
They got exposed to. Yeah, vertebrates. This is the same type of immune system for all vertebrates, especially we're more closely related to mammals, but yeah. But babies cannot get their first vaccines until they're 8 weeks old. So that's why the pediatrician told me that. It's because during those 8 weeks, they have not been vaccinated. I could not breastfeed. And we'll talk about how breastfeeding babies is healthier for a baby because they get passive immunity. They get antibodies to their mother's breast milk. I couldn't do that because of the medicines I take. So my children were way more susceptible to becoming sick. So he was like, don't leave the house. Don't go to Target. Don't go to, he's like, don't leave. And anyone that came to our house that it really had to make sure they cleaned their hands and they weren't sick. Because babies are way more susceptible to becoming sick. And if a newborn gets sick, they're much, because they have no way to defend themselves, really, like, they could die very quickly. So I had to be really careful. And I really didn't leave the house, I think, until 10 weeks with the twins. I mean, I went to the doctor, like, you know, we went to the pediatrician, but that's all we did. And with Marie, I don't think I left. Now, I went on walks and stuff. Like, I didn't take them out in public, is what I mean. Like, I went on really long walks around the neighborhood and all that, but it was just me, and they were outside in the nature, and, like, that's the only way I could get my twins to sleep at the same time, honestly. It was really long, like, 2-hour walks. And it was like but one time I went on a 3-hour walk with Marie. I walked like 9 miles. Something crazy. Because she's fun. It was her house. I'm active, so I have to move. So, you know. Why not? Everybody's like, sleep while the babies are sleeping. I'm like, I'll walk while the babies are sleeping. Or I would run. So anyway.
Oh, they're in the stroller. You're just going about you.
Do you think I
Actually, the dog while they're sleeping.
I thought they were walking with you.
She's just driving.
They can't walk 3 miles from me right now, so. Anyway, so this is something that as you're exposed to pathogens, you develop more and more of this line. Okay, so that's why it's called acquired immunity. It involves cells called lymphocytes, so your B cells and your T cells. We'll talk a little bit more about those in a little bit. And it also involves proteins called antibodies. Antibodies are proteins that are produced and released by B cells. The antibodies will circulate in your fluids, your blood and your lymph, and they basically function to bind to a pathogen and like handcuff it. So it can't cause any more harm until a white blood cell comes and eats it. We'll get back to that in a minute. Yeah, antibodies are also called immunoglobulins. Another name for them. And your third line responds to things called antigens. I have a slide on these in a minute. I'm going to hold on to my, look, I have an antibody.
Oh, quick question.
Here's the antigen. Yeah.
We were talking about like blood types and stuff.
Yeah.
And you said some people have like the RH antigen or something like that. What does that mean again? Like, does that
So an antigen is anything that is like usually a protein. It's a cellular. It's like something that's in the cell membrane, usually of cells. So the RH would be a certain type of like glycoprotein that's in the membrane of red blood cells. Okay? It's not free floating like this.
Okay.
This is just a, a stuffed animal thing. But I have to text the Jay. I think their class got this for me last year. Anyways, okay. So cellular name tags are on, now you have antigens on your own cells. We would call them self antigens. Non-self antigens are what's in on everything else. They could be on the surfaces of pathogens. They could be toxins that are produced by pathogens, things that are secreted by them. We call those exotoxins. Something you respond to, an antigen you respond to that you shouldn't respond to if you have an allergy, like pollen. Okay, pollen is something your immune system, if you have an allergy to it, responds to it abnormally. Pollen is not anything that's going to cause harm to you. Your immune system should ignore it, but for some reason, some people don't. We'll talk about allergies later on. And then antigens are also found on non-self antigens, can also be found on abnormal body cells, cells that are doing something wonky that could become cancerous. And those should be responded to and gotten rid of. They're not always, but they should be. So there's two branches to your active immune system. The humoral branch. When you think of the word humor, you should think of fluids. A long time ago, I don't know why, but they called fluids their humors. And at some point, there were like 4 humors. I don't even know.
Oh, like the thing in your eye, the viscous.
Yeah, kind of like that. But humoral means in your fluids. What kind of fluids? In your blood or your lymph. Okay, so this branch of your immune system, you'll have to kind of listen to me here. So imagine someone sneezed on you with the flu. And the flu got in your nose. I don't have a flu virus. I need a flu virus.
You need the flu and the cold.
Never mind.
He has a cold virus.
You ended up drinking out of someone's drink you had mono. Okay? So, I told you the story about Fletcher having mono?
You said he had it.
I didn't tell you the story? Okay. So, what? No. It was like right when I got pregnant with twins. So I didn't do in vitro or anything, but, I had to go to shots and stuff. And like, I had, at least 2 of them, but I had twins and we were going to the first real doctor's appointment. I went to see a reproductive endocrinologist to get pregnant with the twins. He's a doctor, a fertility doctor basically. And once you get past a certain point of your fertility doctor in your pregnancy, then you go to your regular OBGYN. So I was my first appointment with my regular OBGYN. So we were sitting in the waiting room. I think I was 12 weeks pregnant or something like that. This is an exciting time, my first pregnancy, whatever. And then Fletcher's not feeling good, whatever, it's fine. And then we go and all this stuff happens and everything's fine. But then he starts still not feeling good. And it looks like I'm pregnant for the very first time. He doesn't feel good, whatever. He finally goes to the doctor a couple weeks later. He has really bad mono. It was so bad he took 8 weeks off of school. Some days I would come home, he didn't even get out of the bed. Or he had moved to the couch and he didn't get off the couch. And honestly, I feel like such a bad person because the whole time I'm like, dude, I'm pregnant. I'm supposed to be this person that's everyone dotes on and you're like, you'll do anything for me, meanwhile he's sick. And I get home and I'm like, I can't even go hug him. I can't do anything because the doctor's like, you better not get her mono. So I didn't get near to him. I think he even slept in a different room most nights. It was crazy. For almost my first trimester. He was really, really sick. Which I felt really bad for him. And now looking back on him, that guy was really sick. His spleen was all enlarged. The doctor said he was at risk of having a burst. Put him all these crazy alternative medicines. You can't do anything for a virus. You can't do anything. But they put him all this stuff to try to quell it. It was bad. Anyways, if you've ever had mono, it could be I feel really bad now. But,
Oh my God, get up!
Love me. These are your children.
Usually guys when they're sick are pathetic. I just sort of attributed them to that and so I feel bad about it forever. But you drink after someone and you get mono. So mono is running around in your fluids. Let's imagine it hasn't infected a cell yet. So your immune system wants to get rid of these guys before it gets into any more of your cells. So that's what your humoral side is for. It involves getting rid of the virus or the pathogen before it can actually get inside of your cells. That's the humoral side. Make sense? I still feel bad about that. We're talking about activation is activating the cells involved in here, the B cells. And I'll talk about clonal selection in just a little bit. The humoral side, these B cells are turned on basically and produce a lot of antibodies, and I'll come back to that in a second. The cell-mediated side is sort of the other branch. The other side of your immune system is, okay, right, the way viruses work is they get in your body cells. So the cell-mediated side is the side of your immune system that goes and searches for your body cells that have this inside of it, and it will kill those body cells. That's the cell-mediated side. The cells involved here are called cytotoxic T cells. Cyto means cell, toxic means death. Cytotoxic T cells are your cells that kill your infected body cells. Cytotoxic. So these are different from natural killer cells because natural killer cells are non-specific. They will kill any cell that's infected with any virus. But cytotoxic T cells, once activated, will kill every cell in your body that's infected with only a certain virus. Does that make sense? Yeah.
Okay, in PDF video, is it like you already have like one cytotoxic T cell and like a B cell that are like ready to respond or whatever?
Mm-hmm.
So you have one for like every single virus?
Yeah,
like
crazy. Basically, you have over about 1 billion different types of antibodies in your body.
Like how would they know if they haven't been exposed to it?
They don't, it's random. And it's based on what you do in here. So there's around 300 genes. We'll talk about antibodies and how they're produced. But you have about 300 genes that will code for the different, what we call variable reasonable antibodies. Because through alternative splicing, you produce about 1 billion different types. Now you might have a lot of your body that are never exposed ever. Like they exist for pathogens that doesn't exist. You know what I'm saying? Yeah, it's crazy. But it's inherited. So that's how it works. But you don't know. You just have them. Crazy. Yeah.
So when people die from a virus, is it because they didn't have a cytotoxic T cell, or is it just because?
So it's usually, no, it doesn't. The virus could have caused their body to, the reason you get sick, it takes around, I have some chart, too far away, I have a chart in here, but the reason you get sick is because if someone sneezes on you with the flu, let's say, okay, and you haven't been exposed yet, which is the point of vaccines, but if you haven't been exposed yet and you haven't been vaccinated, those are antibodies.
Oh, never mind. That was the church.
Oh, no, thank you, guys. That virus could be running around your body, and maybe that one B cell is in like a lymph node, like somewhere in your leg, let's say, I don't know. That one that recognizes the virus. But it takes like a week or two before it even finds it. So during that week or two, that virus has multiplied, infected your respiratory tract, you've gotten really sick, whatever. People that die of the flu, their immune system, usually it's called a cytokine storm. So their body is doing all this stuff, and all these cytokines are released like in huge quantities, and then it causes their body to die. Like they stop breathing and all this crazy stuff.
So it's like kind of on a time.
Yeah. Yeah. I mean, eventually it's going to run into that B cell, but how long is it going to take?
Like 24.
If you haven't been exposed, it could take a long time. That's going back to it. Okay. And y'all, I try to answer as best I can in a short amount of time as I can. Like I said, I took a whole class on this, like a whole year. It's a lot of information, yeah. Yeah. Your lymph, that's sort of like plasma. I mean, yeah. Well, it has no, nothing's moving it around per se, except for just the random contraction in the skeletal muscles. It's very slow moving. Okay. So this is a chart, kind of like the one in your, or maybe it is the one in your book, I don't remember at this point, but kind of overviewing the entire immune system. Humoral side, cell-mediated side. And I'm going to kind of talk about all this stuff lots of times as we go through this chapter today. So the B cells, let's stay on the humoral side for now. So here's an antigen that you have been exposed. Someone sneezed on you with the flu, okay? And it's running around in your bloodstream, all right? An antigen can be recognized by a B cell. When that happens, what happens actually? The B cell basically turns on. It's like tag you're it, okay? And it starts running around. Now it doesn't really run. What it does is it starts dividing. Dividing very rapidly and producing lots of clones. That process is something called clonal selection. Sounds weird. It's been tagged and it divides, so clonal selection. It was selected to make clones. But anyway, the cells that are produced will differentiate into two types of cells. Memory cells. The memory cells are not really going to do anything right now. They're just going to hang out until the next time you're exposed. But the point is you have lots of them. Now, like I said, you're born with about 1 billion different types of things, like of antibodies that can respond to about 1 million different types of antigens. But you only have one of each. But after you've been exposed, well, now you have a lot of these that will respond to that particular antigen.
So they respond faster because there's more of them.
Yes. It's much more likely to be run into it. You know what I'm saying? So yes, it'll respond faster because there's a lot more. Yeah.
Okay, something about vaccines, how they develop boosters.
Yeah, vaccines, depends on the type of vaccine. We say usually the max amount of time a vaccine will last around 20 years. But you get a tetanus booster rather than 10 years. The reason you get a flu shot every year, though, is because the strains are different every year. So it's not like your immunity from last year is gone. It's just that the strains could be different.
So like ads.
Yeah,
So how long do memory cells last?
10 to 20 years.
Okay.
The plasma cells are the ones that produce a lot of antibodies. These are the ones that are going to secrete huge quantities of the antibodies. The antibodies run around your bloodstream to tag or handcuff the pathogen to prevent them from causing any more harm. I'll come back to that in a minute. That was the chart you just showed me.
So those are the ones that were pumping the things out in the video.
Yes, these guys are the ones that are pumping the antibodies. Do you see all these lines here? That's like indicative of the ER. Their major job is to make protein for export, so that's why they have a lot of ER. Okay, so that's the B cell side. The cell-mediated side is the same kind of thing-ish. If they respond to an antigen, and these B cells and T cells kind of hang out, the place they like to hang out is your lymph nodes. Your lymph nodes are places that what we call antigen-presenting cells go to show off. I'll talk about that in a minute. Your cytotoxic T cells, if for some reason they are exposed to this antigen, they undergo clonal selection as well. They'll divide, produce a lot of clones. Their clones will make memory cells as well, and then they'll make active cytotoxic T cells. Now, these guys don't make antibodies, but these are like the trained Army Rangers. Now, all of a sudden, instead of just one, you've got like 50 of them. They're all going to go find the same pathogen and kill it. So the same body cell, well, the body cell that's been infected with the same pathogen and kill all of them. Questions so far? Okay. The helper T cells are in the middle. These are the ones, the helper T cells communicate to both sides. If the helper T cell becomes activated itself, it will be like, it'll send chemical signals to the B cells and chemical signals to the T cells and say, both you guys undergo clonal selection and get rid of this pathogen. The helper T cells don't do anything itself. Does that make sense? It just activates both sides. Now, a B cell could activate a helper T cell, and cytotoxic T cells could activate these guys to talk to the other side. This is why HIV is so bad. Because if HIV gets rid of these guys, you have no more communication. So it's sort of like this side could be working, but if you get rid of a virus in the bloodstream, but yet you still have viruses living in your body cells which aren't being killed, it's like you're overworking yourself, but it's not. It's like you're fighting a losing battle. Or if you're killing all the cells, but there's still viruses running around, you know. Like, yeah.
So plasma, B cells are for like the liquid cytotoxic.
Cytotoxic. Yeah. Yeah. All right. So let's talk about how these, a little bit more about antigens. Antigens, like I said, are cellular name tags. And they're proteins or glycoproteins that are on the surface of cells. So we have self-antigens. We talked about this a long time ago. These are like your IDs, okay? The ones that belong. And then we have non-self antigens. The self antigens should cause no response. They should be totally ignored. Should be. If you have an autoimmune disorder, they're not. That's a disorder. Foreign antigens are ones that don't belong, and those should elicit some sort of response from a white blood cell. And they could be all kinds of things. So things that are actually pathogens, viruses, bacteria, protozoa, parasitic worms, like a tapeworm, fungi, toxins, exotoxins, even non-pathogenic stuff, cancer cells, which are abnormal body cells. Pollen, which is something that really shouldn't elicit a response because it's not harmful. What's interesting enough is that transplanted tissue can also cause a response. Because it's not exactly like you, unless it's an identical twin. So if you ever have to have an organ transplant, you will probably have to be on immunosuppressant medication for a very long time, if not the rest of your life. Your body doesn't reject that organ. So I'm going to pause for a minute and jump to something called the MHC. And I'm skipping way ahead in here because I have it somewhere else. But we'll come back. Okay. So, self versus non-self. There's something, so how does this actually work? There's something called the major histocompatibility complex, or MHC, protein. So they're on the surface of every cell in your body. You have 6 different locations in your genome where these genes are located. And if you're diploid, that means you have 12 different alleles. 6 times 2 is 12. So there's 12 different alleles in your body. And in the human population, there's literally hundreds of them possible. So if you take those 2 into account, 6 different locations, if you're diploid, you have 12 different alleles, and there's hundreds of available population. It's impossible for 2 people to be exactly the same. It's highly unlikely, unless you're a twin, identical twins. It's your cellular fingerprint, and it really marks you as highly unique cellularly. Yeah.
Why is it that twins can have like different?
Okay, right. This is a cellular fingerprint. So these fingerprints are different. These actually form in utero and have to do with different stuff, like the way the amniotic fluid is moving around on this crazy stuff. So I can show you videos. Didn't you watch something like that when we would have to tell?
No, we just saw pictures. Miss B was ruthless. She put all the pictures out there. Everything.
She's just trying to scare her.
Yes. It looked like cauliflower.
Oh my God.
Yeah, it was like a frosted pink.
Disgusting. Okay. You're a doctor or something, you might have to see that stuff.
That's why I was sticking.
Yeah, like,
Ain't gotta see no junk though.
geographic.
It's just power. I'll take that. It's gonna be junk.
I have a lot of thoughts right now. Okay. Anyway, so this is how your body recognizes self versus non-self. Now, even twins, because of now what we know about the epigenome, these can even change a little bit in twins. Genetically, they'd be the same, but depending on what's expressed or not, it still could be different in twins. But, okay. What's also interesting about this, I'll talk about it now, is that it's genetic variation is beneficial, right? Like, it's good to be very different. What's crazy is that I have read research. I'm really interested in psychology, and I always have been. So I took a lot of psychology classes in undergrad, with a ton of stuff, and I've just always read stuff about it. Human psychology is crazy because we have the most complicated brains, and we have free will, and we have all this stuff that almost no other organisms really have. Although elephants can cry. We do animal behavior. I'll show you. Elephants mourn when they have another elephant that dies. They have this crazy. They mourn them, and they have a process. They walk around them and process, and they have this little ceremony. They whimper. They can have pain. The elephants fear. Crazy. Anyway, humans are just really different. But what's crazy is I have read research that talks about how females, a lot of times in the animal kingdom, females choose their mates. Females are the major ones that choose. And it makes sense because, honestly, sperm is like a dime a dozen. Males make sperm, and they make so many, and they make it throughout their entire life. It's sort of like sheep, honestly. Eggs are not. The females form with all the eggs she's ever going to have, and then they're there. And so you have to be really picky with who you're fertilizing your eggs with. You know what I'm saying? So females are just really picky. Now, humans it's crazy to think about. You can study it in a lot of other animals, and we talk about that later on when we do evolution and animal behavior, but humans is different. But I've read studies that show that females have a tendency to be more attracted to a mate that smells different, and they can detect it. Now, you don't consciously do it, but females can detect the smells different, and then the different is their MHC is almost entirely different or more different from their own. Does that make sense?
Yeah.
The opposite.
Yeah. But if you think about evolutionary wise, it makes sense because it causes you to have more genetically variant children. It's crazy. But you know how, like, if you ever have a sibling, like, you pick up their shirt and you're like, I don't know why, my sister, like, I don't like the way her shirt, like, she smells weird to me. She doesn't smell bad, but I just don't like the way she smells. It's probably because genetically she's probably the same or very similar. But it's crazy. And, like, you can, like, if you put 1,000 white undershirts in front of me, I will be able to pick out one that's Fletcher wore every single time. Crazy. Like, every time. You can just tell. And, you know, it's just, and you probably, I don't know, all that stuff is really interesting.
Wait, so if you don't like the way it smells, and then like y'all just like use that up, yeah.
I don't know what I smell like, because I don't, you don't know what you smell like. It's weird.
It smells very unique, like it's not as bad as.
I think I have a video on it. Hold on. Yeah, it's this funny t-shirt thing.
Yeah, you can also tell who your sibling is no matter what.
Yeah, I can tell my brother's stink.
What was that, D News? What's that? This is not.
That doesn't look right.
No. Wonder where it is. Go away.
I've never heard
Shouldn't that be the opposite way?
I feel like this is it. Okay, hold on.
You smell. I don't know if
This
you
is
smell
it.
more left than anybody else, but you smell. Anthony here for D-News, and hey, I didn't mean it as an insult, man. I'm sorry. It's totally cool. It's just part of being a living thing. We all smell, and we actually all smell different. It might be hard to tell on a city bus or in a crowded locker room, but your brain definitely picks up on it. So much so that you tend to favor people and things that smell differently than you do. See, we're all covered in these molecules called major histocompatibility complex proteins. Animals use MHC peptides to find a mate, and scientists have found out that we might actually use them too. See, you've probably heard of pheromone parties. You heard of these things? This is a party where everybody brings a sweaty shirt in a plastic bag, and then everyone passes around the plastic bags and smells them, and whatever shirt you like the smell of the most is supposedly somebody you'd be a good romantic match with.
That is not, that's weird.
There was a 1995 study using sweaty shirts that showed that women prefer people with a particular MHC scent. So, are they looking for certain kinds of things? What kind of MHC scent appeals to the ladies? Well, don't worry about not having the kind of scent that your one true love is going to be into. It turns out that we're not all that sensitive. We're just basically looking for something that doesn't smell like us. As a follow-up to the stinky shirt study, scientists tested different scents and perfumes on people, and some of them had their own MHCs mixed in, and the ones with their own MHCs triggered a response in one region of the brain, And all the different smelling MHCs trigger another one. The idea here is that you're probably choosing perfumes or deodorants for yourself that are amplifying and working with your own subtle smells to make them stronger, but you won't like that scent on a potential mate. So how do you make this work for you, other than having to go to a sweaty t-shirt party? Well, you're probably creepily sniffing your crush anyway. Just take note of the scent and then try to wear something that smells as different from it as possible. And do not get caught smelling them, for God's sake. I have made that mistake one time. Have you noticed a particular smell that irritates you? I'm kind of into vanilla. Does that mean I smell like chocolate? Subscribe, and we will see you tomorrow.
Sorry. I forgot about that video. Okay. Anyway, kind of crazy, right?
Can we still see the elephant cry?
Not today. You will. You will. Okay. So how do these actually work? So MHCs, the way they work is they're proteins. So the MHCs are, Chelsea said they're lilac. So these are the lilac MHCs. They originate on the inside of your cells. It's on all of your body cells. What they do is they will basically grab a random protein. It's on the inside of your cells, in the cytosol. And they will migrate to the cell membrane and then display that protein sort of like a flag. And it's like a snapshot of what's going on inside of your body cell at any given time. And if the proteins being displayed are normal, then it'll be ignored by your immune system. So if the proteins being displayed are abnormal because you're infected with a virus, or your body cells are abnormal because they're cancerous, okay, it should elicit a response from your immune system. So that's actually how the MHCs actually work. Yeah.
So is that why some people kind of like to smell sick?
Yeah.
I work in a hospital, so I doesn't smell sick. But it hurts.
Oh, probably.
I don't know.
I don't know. I don't know.
They
I don't know.
like they just smell like that.
Okay, I wouldn't say they smell like death, but I do get that, like, I feel like if someone's sick, I'll be like,
Probably. Probably. It should be true to what's going on inside the cell, but if it's like transplanted tissue, for example, well, the actual MHCs themselves will be different. But so we share some genes in common, some proteins in common with bacteria. You know what I'm saying? Because we have we undergo glycolysis. I mean, you know, so it could be similar, but you know what I'm saying? Could be. But the collection of a lot of them, however many are genes displayed, is probably likely that it's unlikely that they all would be the same. All would be the same. Does that make sense? Yeah. Yeah. It wouldn't cause any problems. Yeah, it wouldn't matter because it's all the same body. Okay. So there's two types of MHCs, class 1 and class 2. Yeah. Okay, yeah. Did you look it up? Yeah, it's crazy, right? Ooh. I hate hospitals. That's why I never get I know. So if you
just go to the hospital and walk around. No one can say anything.
So your derm, so you have epidermis. Your dermis is like, now you're capillary.
Like, I'm a nurse now.
I can stick my finger out before it's dead.
And all of this you're looking at is that tiny layers of ectoderm have in my pocket. They're dividing and keep pushing up and cells die. When you get a burn, there could be burn, you get rid of your epidermis and your dermis. So there are no one of those epidermal cells to replace, you know what I'm saying? It's not there anymore. So that's how a transplant works. You take a full layer of the stem, the dermis, and the epidermis, and then the little layer of cells in between, and you put it.
So wherever you take it off, usually get all like.
Yeah, so one of the things they do is they try to, they put like a water in there, like a place on your back or something, and you slowly inflate it, so that the animals are always growing, they giant bubble of extra. They take it off, and
Oh,
then.
that's like that guy that's throwing glue on his nose or something onto like, I think your nose on his face, like tomato. I think when they were talking about like that, and I took it off.
Yeah, but they just sort of skin back together. It's not like they cut off the skin, you can do that when it's dry.
Yeah.
So they grow extra skin and then take the extra off and just, you know what I'm saying? Connective tissue. The way the type of connective tissue that makes up scar tissue would actually.
And we have to get this the way they attach her dog to the leg, like,
Right. I know, okay. Anyway, you can smell sick, I need to add that in my notes. Weird, I hate hospitals, that's probably why. Alright, class 1 MHC molecules, you have to follow me here. There's two types of MHC molecules. Class 1 are found on almost all of your body cells. Your liver cells, your skin cells, your heart cells, whatever, almost all your body cells. Class 1 MHC. And they will present antigen, like a snapshot of what's going on inside of your body cells at any given time. As long as they're normal, the cytotoxic T cells are the ones that bind to them. They will leave the cell alone. The way the cytotoxic T cells bind is they have, and it's not shown on here, but they have a protein component, looks like that kind of, called CD8. The CD8 protein, this one cytotoxic T cells, will bind to the class 1 MHC and kind of hold it there, okay? As long as the cytotoxic T cell recognizes this antigen as self, it'll leave it alone. But if it recognizes it as non-self, what do you think that cell is going to do? The cytotoxic T cell will kill the cell. It'll start releasing a protein called perforin.
like way away.
Pretty good. Not, this PowerPoint is kind of all over the place. I just
Oh,
thought
it's so little compared to that picture of it. It's giving it its all.
I know. Here, hold on. Oh, there. Okay. So when it recognizes that there's our CD8, at Torin, it'll start releasing chemicals. These chemicals are called perforin. Perforin will poke holes in your infected body cell, your abnormal body cell, and cause it to lyse. And it will also induce apoptosis in that cell. Not only poke holes, it'll cause it to start bleeding, and it will die. Yes. Okay. So that's what happens. These are the class 1 MHCs. Questions? We'll move on to class 2. Okay. Class 2 MHCs. I need to reorganize the PowerPoint. There we go. Class 2 MHCs are found on, write this down. They're found on antigen-presenting cells, APCs. Antigen-presenting cells are dendritic cells, macrophages, basically. These cells function to eat foreign invaders. When they do them, they digest them. So they're supposed to be. And then they present their foreign protein. That's their job, is to present antigens. That could be foreign. To tell the body, hey, I just ate this guy that doesn't belong. You need to do something about it. Okay? Do we want to kill this antigen-presenting cell for displaying something foreign? No, it's not infected. We want to leave it alone. So it presents its antigens to helper T cells. The helper T cells, if they recognize it as foreign, and by the way, their little protein that helps bond is called CD4. Have we seen that before? Where HIV will bind. So if it recognizes it as foreign, the helper T cell doesn't kill the APC. The helper T cell will then go activate the humoral and the cell-mediated side. Be like, hey, there's this foreign pathogen that could be floating around in our bloodstream and we're maybe even infected our body cells. You need to go find them and kill them. Make sense? Yeah.
Which are the 2 classes?
They're both effective for different reasons. So the class 1 ones are effective because they will be showing like, hey, I'm infected. I need to be taken care of. Like, I need to kill me. So you get rid of your infected body cells. You know what I'm saying? And then these can alert the immune system of a possible infection. Your dendritic cells, major role, they will hang out in tissues, eat invaders, and then they'll go to your lymph nodes. That's what they do. They eat stuff and they go to your lymph nodes. Your lymph nodes are where your B cells and T cells, they hang out and have a little party. And they wait on dendritic cells to show them something foreign to become activated. So they're both really effective for different reasons. Did you say something?
I also want to know if dendritic cells have something to do with hers.
Dendrites.
With dendrites? No. Dendrites are our neurons. And
Yeah, okay, yeah.
Okay. Bless you. So, this is a picture of helper T cells. No. Where's our helper T cells? Here it is. Okay. So when a helper T cell binds to a class 2 MHC, there's our CD4, it won't kill our antigen-presenting cells. What it does is it releases chemicals. It releases cytokines that will induce division in your cell-mediated and humoral response side. It also releases interleukin 2 to help itself or stimulate other helper T cells. So, yay for the MHC. So we have to rewind a little bit. Any questions? I think this is a picture in your book. Same thing.
Wait, it signals what to divide? The
Helper T cells will signal B cells and cytotoxic T cells to undergo what we call clonal selection. I'm gonna rewind. We need to talk about antibodies. All right, antibodies.
All right, I think we're back. No, we're not.
We went back.
Oh, yeah.
We went back.
It's right here, look at.
I'm taking a learn, I'm sorry. Antibodies, remember, are the proteins that are released by what? B cells. Okay, so this is the structure of an antibody. An antibody is just a protein. They're called immunoglobulins. They have 2 different chains, what we call a heavy chain, which is the long purple ones here, and a light chain, just because they're shorter. See the C's on here? C means constant, all right? And then B means variable. You have around 300 genes that will code for different antibodies, but you have about 1 billion different types of antibodies. Why are they can be so different? Because you have 300 different genes, and through alternative splicing, you can produce all kinds of different combinations in these V regions. These V regions are where the antigen will bind to, the antigen binding site. The antigen will fit into these variable regions, sort of like a lock and key or like an enzyme and substrate. It has a specific molecular fit. It will only bind one place. Now, antigens can have different regions on like one antigen, different areas that different antibodies can actually bind. We call an antigen binding region or region on an antigen that an antibody will bind an epitope. So it's on the slide somewhere, but an epitope is one place on the antigen that one antibody can bind. Yeah. So on my little guide here, the antigen binding sites are right here. So here's my antigen. This guy can have like 4 different epitopes, 4 different places that different antigens can bind. The epitope on here happens just to be the bottom. It will bind to this guy, see how it fits? Right? But it doesn't really fit this side. But a different antibody will fit on that side. Does that make sense? One antigen can have different epitopes for different binding sites, but like different antibodies can fit. This one in particular fits. He's tagged. Okay. What's your favorite? They all have different stories.
You can't ever ask her that question with like children.
No, they all have different
I'm
stories.
sure my parents have a favorite child.
Colin and Keisha. I don't even know who named her. I've had her since my very first year.
I love Keisha.
Hmm? My
My
monkey. My very first year teaching, I taught a biology class, and in that class, this is 15 years ago, I assigned like scientists of the like famous biologists. They had to dress up like one and give a presentation. And one of my students said Jane Goodall. And she brought that from her house because she was like Jane Goodall. So she just gave it to me. So I had it for 15 years, and I think it was the year I had George. I don't know when she got her name, but she didn't say her many years ago. That T-shirt, I'm pretty sure is Jordan Stifler. So. I don't know. She used to, people, one year, a different, like, she would hang out at someone's desk every day. Someone would just pick her up and sit with her. That was weird. Anyway, that's her. I don't know, they all had different stories. Like I said, this one came from a James class. Hervey's came from Sunday's class. I don't have the flu, and my rhinovirus is taken.
And your birthday is what, March 12th? 6th. I know it was something around there.
Oh,
I did.
oh,
Oh, that's next Monday. I mean, no, 2 Mondays from now.
It's a Tuesday.
Tuesday. Hey, he is it now.
I'll be 29.
And a quarter.
I don't know, my mom's always like 21.
I don't feel
Oh, that's okay. No, you got 1 more year till the big.
I just keep trying to run a faster mile than I hope to. The other day I ran at 7:30. I about died. All right, anyway. That's what anybody looks like on a computer generated model. So, what do they actually do? So when an antibody binds an antigen, they can do different things. In general, their major goal is to prevent the antigen or the parasite or the pathogen, whatever, from causing any more harm. So they bind to it, render it unable to cause harm until a white blood cell can come and get rid of it. So that's what all these things are doing over here. So they will bind to the pathogens, prevent them from causing any more damage until a white blood cell can go eat it. Same thing here. They could cause some toxins to precipitate out of your bloodstream until white blood cell can eat it. Sometimes the antibodies will cause your complement system to become activated, which will then poke holes in the parasite and then cause it to die. Yeah.
What's the difference between a pathogen and
So a pathogen is whatever the organism is or virus. The antigen are the proteins that are on the pathogen. Make sense?
okay, so like the virus.
One pathogen can have many different types of antigens.
And it only has to get one of the antigens.
Yeah.
Something.
Yeah.
But it can bind, why does it bind to more than just one? Like those.
It's probably likely that if you are infected with a bacterium, you activate more than one type of like antibody. Does that make sense?
And so it's just attractive, right?
Mhm. Whenever the antibodies have to be produced, these cells have to divide to make the plasma cells that will secrete them, but then they just float around in your bloodstream. Whenever they come into contact with it, they'll like bind to it. Okay. Okay.
I was stressing that.
There's different types of antibodies. We call, again, they're immunoglobulins. So IgM immunoglobulins we're involved in what we call our first immune response. Okay, this picture here. So here's antibody level. These are weeks. This is like day of exposure. Okay, when you're exposed to the antigen time 0, the first antibodies that will be produced in your bloodstream are what we call IgM. The second ones that will be more long-lasting over a longer period of time are the IgG. Anyways, the IgM ones also are the ones that activate the complement system to attempt to poke holes in whatever the pathogen is, if that's possible. The IgG antibodies will also increase phagocytosis. Because they're longer lasting, this will be the major one probably floating around your body at any given time. IgA antibodies are the ones that are found in external secretions like milk or sweat. So it's good to breastfeed. We'll talk about that. It's called passive immunity. IgE antibodies are the ones that are involved in allergies. So they release a lot of histamine. Anybody have allergies? Yeah. What do you take? You take antihistamines. The histamine release in your body is what causes you to have an itchy nose, the runny eyes, the runny nose, the sneezing. All that is caused by histamine release. What? Benadryl and histamine. Yeah. So when you take these things, yeah.
Knock you out.
No, you don't have to know these at all. It's just interesting. Go to immunology, college, you have to know way more about this. I just think it's interesting. Oh, sorry. You don't need to know. This is about the first thing I've talked about that you don't need to know. Everything else is important. It's just interesting. Okay, now B cells, they have the receptors stuck in the cell membrane of B cells look like antibodies. I mean, they are antibodies, but they're like stuck in the cell membrane. That's what they look like, just FYI. So a B cell, if a B cell runs into an antigen and binds it, it can activate itself and start undergoing clonal selection, okay? But anyway, it's like stuck in the cell membrane of B cells. And they're called membrane antibodies or membrane immunoglobulins. Anyway. So, are you pretty good with the humoral side of our immune system? I feel like I've talked about that a lot. Here's the humoral side. You're exposed to an antigen, your B cell will bind to it on one of these little things here. Once that happens, what happens in your body? This guy starts dividing very rapidly, producing clones. Those clones will differentiate into memory cells.
Plasma cells.
And plasma cells.
Those plasma cells secrete antibodies, shoots them out the cannons.
We'll bind these guys and prevent them from doing anything until like let's release them.
Yeah.
This is just tomorrow. We haven't gotten to cell mediated.
I'm gonna be like
Yeah.
baby test and all.
We did both.
Plasma cells are also called effector cells because they carry out the effects of the humoral side. They secrete the antibodies. I sort of talked about cytotoxic side a little bit. Don't, yeah. The problem is it all overlaps. Because they make cheese, I'll talk about kind of those, a little, you know.
Oh, y'all do have the same thing.
Yeah.
Oh, thank you.
Okay, vaccines. We're going to kind of take a pause and talk about vaccines for now and then watch the video. We might have to be done after this. So, vaccines are when you expose your immune system to a harmless derivative of the pathogen, mainly viruses. However, you can be vaccinated against some bacterial infections as well. So, it's harmless, so it's non-infective. It won't cause you to become ill. I mean, there are a few live vaccines out there, but not many. It will cause your humoral response to produce antibodies to the pathogen. Why won't it activate your cytotoxic T cells? It's not live. It's not going to actually cause you to become infected, right? So it really kind of works on this side over here. The point of the vaccine is not really to make antibodies. The point is to be what? To make your memory cells. That's the point. Right? So, and then the next time you come to be exposed, you have all these memory cells. It's much more likely that you'll run into the pathogen at a quicker time, and then you won't get sick or you won't get as sick. This is a graph that you need to know. So, I'll show it to you. I'll talk about it. Can everybody see that? Or is it better here? Better there. Okay. Same graph. It's in your book. Over here is amount of antibodies or concentration in your blood, right? And over here is day. Time of exposure is time 0. When you're first exposed to antigen A, right? Someone sneezes on you. Notice your antibodies actually aren't even being produced in your body until around day 6. Why not? What's going on in here? It hasn't found it yet. It hasn't even run into a B cell, right? Or it just hasn't run into anything. So during that time, that's when you could get sick, you know, with the flu or whatever. But at the time it runs into it, doesn't take that long, but your antibodies start to increase pretty dramatically in your bloodstream, and then eventually it goes down. You're probably recovered in here. Now notice exposure, second exposure to antigen A, here. Within about 24 hours, what happens to the antibody level of that? It goes way up, like twice or 3 times higher than before, because you probably activated more memory cells, and then they start secreting plasma, producing plasma cells, making antibodies at a much faster rate, which is why you don't get sick or you don't get as sick. Right, and here they're showing another exposure, but to a different antigen here, antigen B, and it's the same response. The point of this graph is showing that every time you're first exposed to an antigen, it takes like this long, and then the same thing for the restarts.
So it can be like a different strain.
Could be, yeah. Last year, Miss Hatter got both flu viruses. Like she got infected with 2 strains of flu
At the same time?
one right after the other.
Oh.
But Jonas Salk is the person who developed the first vaccine against polio. Polio was very deadly and debilitating and attacks your motor neurons. And okay, you don't need to know this, but it's interesting.
Interesting.
Poliomyelitis is the polio virus. It enters the body through your mouth. It will multiply in your intestines and get into your nervous system, and it can cause total paralysis within just a few hours. Yeah. 1 in 200 infections will lead to irreversible paralysis. Among those who are paralyzed, 5 to 10% die. The reason they die is because their intercostal muscles and their diaphragm won't work anymore. They're paralyzed, they can't breathe. If you can't breathe, you die.
Wait, where's the real like sound?
Well, it's not now. Huh?
Where's the sound?
You like that?
Whatever. Polio mainly affected children under the age of 5. So he produced this called the Salk vaccine, and it was based on poliovirus grown in a monkey. Anyways, there's also something called the Sabian vaccine for polio, which was developed in 1962. It used live but weakened, we call attenuated virus. So anyway, polio has been eradicated. It was eradicated in the United States in, 1979 was when the last case in the United States was reported. In 2002, Europe was certified polio-free. Franklin FDR had it. In 1921, and in 1932 was when he was elected president. So the worst epidemic was in 1952. 57,628 cases were reported. So a lot of these pictures, like I said, affected children One of the major ways they help the children breathe is through using something called an iron lung. These images were from Boston Children's Hospital. I'm going to show you a video about this in just a minute, where they had a really bad epidemic. And the video I'll show you talks about how the iron lung works. But if your muscles are paralyzed, you cannot move the air, so you die. So the way the iron lung works is creating external pressure changes that cause them to be able to suck air in and out of their mouth and their nose without them actually moving their muscles themselves. Yeah. No. Most children were in an iron lung for maybe a week. Their body will recover from the viral infection. So the problem with polio is if it infected their neurons, its neurons don't regenerate. So they have permanent, that's why they, this kind of stuff, that's neurons damaged. So they can't activate their muscles anymore, and so they won't, But no, they won't have to. Most children were not on this. And the video even talks about the lady was like, some people were only in it at night, some people were only on it a few hours at a time, didn't have to be in it all the time.
Okay, so for a person that has polio, when the vaccine was developed, do they still get it even if they already had it?
Probably not. Because if you're exposed to the virus and if you already had it, then you have the memory cells, so you won't get it again. Is that what you mean? 1994 is when all the Americas were designated polio-free, North and South. So we don't vaccinate against polio any longer. Hold on. Where? Where? There we are.
That was weird. Why did she make us sing that?
Oh, I don't know. I don't know that.
Where's my polio?
She was thinking because not everyone died.
Oh, yeah. I'm pretty sick.
Mark Rocker.
Poliomyelitis, commonly referred to as polio, is a frightening contagious viral disease that can have devastating effects on the central nervous system. Children are most often affected, but adults can also be vulnerable, as seen when future President Franklin Delano Roosevelt became infected in 1921 at the age of 39. Though this illness has likely been around for millennia, it became more prevalent in the early to mid-1900s as large epidemics occurred around the world. Ironically, these often happened in developed nations, including the United States, as improved sanitation led to reduced naturally acquired immunity. Many children who were infected.
Did you ask how it was? What did you ask? Contaminated food or water, because that's coming to you through the mouth. So.
How does people spread it? Like how does that water?
How do they get contaminated? Let's see. I think it's kind of gross. It would probably be through like stool or something. Just like
Dirty water.
dirty water. Yeah. It spreads through contact with the stool of an infected person or droplets from a sneeze or cough. If you get stool or droplets from an infected person on your hands and you touch your mouth, you can get infected. If your child puts objects, children, a lot of times very young children put everything in their mouth. I mean, they do, little kids. So if your child puts objects like toys that have stool or droplets on them into their mouth, they can get infected. If you think about it, kids are dirty, y'all. They don't know. Like, half the time I go and kids will scratch their bums because it itches and they just scratch, and I'm like, go wash your hands. They don't know. They have to be taught, so that's why. Anyway.
Infected developed a fetus and soon were unable to move their limbs. Some had such extensive involvement of their spinal cord that they also could not breathe effectively. When this occurred, death often resulted from respiratory failure. For many, little other than comfort measures were available for treatment. However, Phyllis Drinker, an engineer at the Harvard School of Public Health, developed a simpler mechanical ventilator that could be used to provide effective respiration for individuals who were too weak to breathe on their own. This large device, which because of its construction became known as an iron lung, was first used to treat an 8-year-old girl with polio in 1928 at Boston Children's Hospital, adjacent to the Harvard School of Public Health. Soon thereafter, iron lungs were being mass-produced and used to treat polio patients around the world. In the early 1950s, during the last large polio epidemics that occurred, much of Boston Children's Hospital was devoted to treating polio victims. However, due to the pioneering research work of John Enders, a microbiologist at Boston Children's Hospital, and his colleagues at the hospital, techniques were developed to culture the polio virus in the laboratory. This enabled Dr. Salk and Sabin to develop vaccines that rapidly led to the eradication of this deadly disease. And in 1954, Drs. Enders, Weller, and Robbins received the Nobel Prize in Medicine for their work. By the 1980s, iron lungs were virtually obsolete, having been replaced by much smaller and less cumbersome mechanical ventilators that are now used to treat patients with respiratory failure from other causes. In order to appreciate how an iron lung functions, the Archives program at Boston Children's Hospital has restored an old iron lung and created this short video.
This is a restored Emerson model iron lung, originally manufactured in Cambridge, Massachusetts.
An iron lung is, I think, aptly named because it is quite a heavy and somewhat unwieldy device. It is made out of iron parts, and it is very bulky and large, and it's very difficult to move around. It often takes 2 or sometimes 3 people to push these big devices.
A patient with respiratory failure was placed inside the device by opening the front end and sliding out a small bed. The bed was reinserted into the machine, and the device closed and sealed. A collar was then secured around the patient's neck to establish an airtight seal.
Depending upon the strength of their respiratory muscles, some of them had to be in there 24 hours. Some were in at night only. Some were in for 2 or 3 hours at a stretch, and then they would be out for half an hour.
Boston Children's Hospital even developed a room-sized iron lung that could accommodate several patients at one time, as seen here with nurses inside and outside the room. The iron lung worked by generating negative pressure within the device when a large diaphragm at the far end of the machine pulled back. This would draw air into the patient's lungs via the mouth and nose, which were extended outside the machine. By rhythmically moving the diaphragm of the iron lung, negative pressure could be generated, simulating natural respiration. The rate of ventilation and the extent of negative pressure generated by the machine were controlled with adjustable knobs. The device was powered by electricity, but could be operated manually during a power failure.
Smart engineers back then thought that there should be a mechanism to have these iron lungs operate. So there's actually a very large lever that you can pull back and forth so that you can physically move the bellows in and out of the iron lung to create the negative pressure. It also has a place for your foot because, again, to really generate the pressure that the patient might need, you would have to use a fair amount of force. So you have to have your foot and your upper body strength to do that.
Operating the diaphragm by hand took much work and was tiring. Hospital staff were trained to relieve each other every few minutes in order for the patient to effectively ventilate. It could be many hours to facilitate unimpeded ventilation.
When you got into the rhythm, you sort of counted the rhythm, it was counted, and you just picked it up and you continued to pump.
During this time, limited access to the patient's body was possible through portals in the side of the iron lung. The portals maintained an airtight seal after a caregiver's hands were inserted. This enabled nurses to keep the patient clean and comfortable. Physical therapists prevented patients from developing muscle contractures in weak limbs. To To see how negative pressure mechanical ventilation works, one merely needs to observe a patient in an iron lung trying to speak. When the machine's diaphragm extends to create negative pressure within the iron lung, air is sucked into the patient's lungs and the patient is unable to speak. Then, as the patient exhales, sound can be produced. Here you can hear a healthy volunteer inside an iron lung counting while the iron lung is operational. Okay, let's see you count to 20. That's just
2, 3, 5, 6, 8, 9.
Note, vocalization is only effective during that part of the ventilator cycle when exhalation occurs.
Iron lungs did their work back when technology wasn't available or more sophisticated. We certainly didn't have the microchip back then. And this was really the best way to support patients' respiratory function.
Fortunately, the widespread utilization of polio vaccines since 1965 has resulted in the disappearance of polio in all but a few remote, underdeveloped regions of the world. Families no longer have to fear their children will suddenly be affected by this dreadful disease, and the few remaining iron lungs are now medical artifacts.
Please help us improve the content by providing us with some feedback.
Looks like they have another system for it now instead of iron lungs.
Well, it's been eradicated. You were still vaccinated against polio when you were a little kid. And the only time you need them, I don't think they recommend getting any more shots unless you're going to travel to a country where polio is still active. So you were given, I think, 4 months. You were 2 months old. I think you could maybe be 4 months old, 6 months old, something like that. It was a series of shots you were given as a child. I think your last was given around between the ages of 4 and 6. That's how a lot of times they work.
The whole thing that you were talking about how we shouldn't get one of the vaccines.
Yeah. Hepatitis, I think, is a series of a couple kind of things. But you were given them a long time ago.
The spike, what was the whole thing about not getting a specific vaccine? That even worse. I thought you said not to get one.
You're given. No, you are. I don't know what I said. You're not vaccinated against polio.
No, I thought there was.
We don't use the oral one anymore.
I thought you said that there was a vaccine that for a specific disease that we shouldn't get or something.
We're not giving smallpox anymore.
Well,
I don't know. Yeah, I know that there's an oral vaccine for polio, but it's not approved in our country. The other countries they give it still. I think it's just live. All right, I'll see you tomorrow.
I know not.